Wonderful lecture, Doctor. Thank you for being here today. Moving on to our next speaker, she is a biomedical engineering professor at the University of Manitoba. Her area of studies is based around the treatment of Alzheimer's disease, utilizing non-medication methods such as, for example, of Alzheimer's disease used for repetitive transcranial magnetic stimulation. Please welcome Professor Zahra Moussavi and her splendid research on the matter.
Can you hear me now?
Yes, yes.
Okay, so start again.
Yes, please.
Do you see my screen?
Yes, yes. Can you hear me as well?
Yes. Thank you.
Okay, good luck.
As I was saying, I have echo. Okay, as I was saying, Alzheimer's disease, the way I describe, is a thief. It comes and steals the most precious memories that people identify themselves with. It is a very clever thief indeed, because people who are affected, they are feeling lost in time and space. The picture that you see on this screen, on the left side, it is the MRI image of a healthy brain compared to the MRI image of a very advanced stage Alzheimer's. As you can see, there are a lot of difference, but at the same time, I also should say that you won't see this much differences in an early stage. This is quite an advanced stage of Alzheimer's disease brain. The changes that you see are especially that the volume of the brain is shrinks, is less than a normal brain. And one of the main area that is being affected is hippocampus. Hippocampus is known to be the source for converting our short-term memory to long-term memory. And also the ventricles that are around in here, you can see that are enlarged. And this picture shows the healthy cells, neurons, compared to the Alzheimer's neurons. And again, the number of the neurons decreases. That's why that the volume in general decrease. Synapses are being destroyed. And the culprit for such changes are the plaques and clusters of beta amyloid and tau protein that clusters around the cells and destroy the synapses. And once a cell, a neuron, is being isolated, eventually it dies. Keep in mind that neurons die in isolation. When they remain idle, they are being died. The same works for humans too. If we are totally isolated, even in this circumstance that we are all supposed to do self-isolation, we connect with each other through the internet. Otherwise, humans being cannot live in absolute isolation. The same applies to the neurons too. Another way to look at what happens in the brain and connect is to consider all the synapses as communication channels, which the neurons basically communicate, talk to each other through the synapses, through the 0 and 1, basically, on and off of the signals that they say. And what the beta amyloid and tau protein does is that it destroys these communication channels, and therefore it leads to the neuron isolation and eventually to the neuronal death. So what we can do for Alzheimer's treatment? There is no cure, but there are some treatments. For example, there are medication treatments. The most famous one is Aricept, acetylcholinesterase inhibitors, basically. But the success rate of these treatments are very, very low. Usually about 1 in 40 lucky people can have a drastic response to the Aricept. The majority of people, therefore, who take it, they show no response at all. There are also non-medication treatments, like neurocognitive treatment, which in my lab I apply that plus transcranial alternative current stimulation, and as well as repetitive transcranial magnetic stimulation, or RTMS for short, which today I'm going to talk about this one. I do offer both of these two types of non-medical treatments, but I'm just going to focus on RTMS treatment for now. This is my lab, and it shows one of our very good patients that I have permission to share his picture. On the left, you see a computer screen that shows the MRI image of the same patient, and we use that one as a GPS for the coil that we are going to deliver the treatment. This is the coil, magnetic coil, and behind me is the TMS machine. TMS machine is basically these two together. The technology of it is pretty simple. It's large capacitors. It creates an electromagnetic field. If you're wondering about the intensity of the field, compared to an MRI machine that is typically about 3 Tesla, TMS machine creates about 0.5 to 1 or maximum 1.5 Tesla. The magnetic coil, we hold it over the scalp, and when we give pulses, each pulse induces a current as a result of electromagnetic field that creates. Okay, this is for a simulation of the coil. We use a figure 8 coil. When the winding of the coil is like the way it is shown, like a figure 8, the intensity is in the center of the coil. So we try to locate the center of the coil over the area of the brain that we aim to stimulate. Okay, back to this picture again. What TMS does is that, as I said, it generates a magnetic field, therefore it induces an electric current inside the brain. It doesn't give electric current, it induces electric current in the brain. That current can activate the neuron, depolarize, or hyperpolarize. It depends on the frequency of the pulses. If the frequency is low, let's say 1 to 5 hertz, around that, it causes hyperpolarization. That protocol is used usually for depression treatment. If the frequency of the pulses are high, more than 10 hertz, it depolarizes, so it is excitatory effect. We did a pilot study. That was the very first pilot study in Canada and US. At the very beginning, I ran it in 2013, quite 7 years ago. And the reason that I'm presenting that result first, because I'm running right now a large clinical trial, but I can't give the detailed result at the moment because it's ongoing. In my pilot study, we had 10 participants at different stages of Alzheimer's, and we divided them in two groups. Half of them received active treatment first and then sham, and the other half received sham first and then active treatment. This is called a crossover clinical trial design. So all of the participants received sham and active treatment eventually. The protocol was 4 weeks of treatment. We gave the first 2 weeks was every day, third week was 3 times a week, and last week, the week 4, was only once. This is a little bit of a result of their MoCA score. Is a cognitive scale score. Blue is people who received active treatment, and orange is the score of people who received the sham. The higher is the better. We subtracted the baseline, so if baseline week 0 is the 0, it shows that the people who received the real treatment, they improved. And statistically, it was significant on week 3. This graph shows the improvement for people who were at earliest stage of Alzheimer's. As I said, we had different people at different level of the Alzheimer's. Overall, I can say that people who were the earlier the better, basically, to show a positive response to the treatment. That's given in every disease. It is like that. When you diagnose a person with a neurodegenerative disease, if you apply treatment at earlier stage, it is much more effective. But no treatment is a cure. So TMS, good positive results can also last just temporary for a short period of time. I would say between 1 to 3 months maximum. After 3 months, we have to repeat the treatment. Currently, our protocol is a little bit different. It is a multi-center international study between Canada and the US. Between Canada and Australia. It is a randomized, double-blind, placebo-controlled study. It means that people don't know if they are receiving sham or active treatment. Our assessors don't know it either. The caregiver don't know it. And we have 2 groups of active treatment and their corresponding sham treatment. The sham treatment is given by the sham coil, which looks exactly the same as real coil, but it doesn't really induce any current in the brain. The protocol is either 2 weeks or 4 weeks every day. We assess people at baseline, then post-intervention, then every 2 months post-intervention up to 6 months. And there are a bunch of standard questionnaires like ADAS-Cog and verbal fluency, caregiver questionnaire, and so on. And of course, everybody who receives sham after the period of the study, we let them know, and they can receive the real treatment. The parameters of the TMS treatment is important. The most perhaps important parameters are, one is the frequency of the pulse. We give them at 20 hertz to be excitatory. Number of pulses, currently we give 1,500 pulses. Before I was giving 4,000. Even right now, for some other studies, I give 4,000 pulses. If you ask me, I'm not 100% sure which one is better. We have to wait until till the end of the study and see which one are better. But it seems more or less they result in similar. And the other important parameter of the RTMS protocols is where we stimulate. We apply to dorsolateral prefrontal cortex bilaterally. Why? Because to put it in very simple words, although I wrote a little bit about neurophysiology of the DLPFC, but in very simple words, DLPFC area is the hub station of the brain. It has many projection to all other parts of the brain. And when we apply to DLPFC, basically we increase the glutamate-glutamine cycle and restore glutamate cycle to the normal. It increases cerebral blood flow and glucose metabolism that has been shown decreases in Alzheimer's brain. And therefore, it enhances the synaptic plasticity, and that's our goal, that we stimulate the neurons, and that way we can boost the brain plasticity so that patients' cognitive status improves a little bit. And this slide is all about why we choose dorsolateral prefrontal cortex. I'm not too sure about the neurophysiology background audience. For the sake of time, I can skip this one. And if you have any question, please let me know. One thing, however, I would like to mention on this slide is that the positive results of high-frequency RTMS treatment also may provide a pathway for future therapeutic intervention. For example, it might be more beneficial that we give it a medication, but at the same time combine it with RTMS treatment, because it can provide a pathway for the medication to be more effective. This is something that my colleagues in Italy are looking into that, and we are hoping to start a clinical trial for it too. Who is eligible to participate in clinical trials that we run? Usually people at early and moderate stage of Alzheimer's, and people who have Alzheimer's or have a diagnosis of mixed Alzheimer's. And they have to be between 55 to 95 years old and being on a stable dose of medication. And remember that I compared the TMS to MRI. So whoever who is not safe to have an MRI will not be safe to receive TMS treatment as well. So people who have pacemaker are not eligible, as well as people who have had a history of seizure, because TMS doesn't have much side effect, but one of the serious side effects could be seizure for people who have a history of epilepsy or seizure. This is a video. I'm not going to show it for the sake of time, but you can Google my name and the term RTMS, and it shows up, and you can see the video for the protocol of the treatment. Overall, all I can say is that TMS is effective about on 75 patients, and out of those 75% of people, about 40% of them respond significantly positive to RTMS. But keep in mind that I'm saying the result has to be repeated every 3 or 4 months. And these results compared to the medication treatment response, which is about 2%, 2.5%, well, it seems to be a little bit better, at least. I'm going to stop here. We also are working on being able to predict the response of the patients to TMS at baseline. So because not everybody is responsive to TMS, and if we can predict it, perhaps we can optimize the treatment protocol. And that is the use of electrovestibulography, who my colleague is going to speak after me. But this is a work on investigation. I would like to thank NSERC Riverview Health Center, private donors, and Western Brain Institute as the funding for the studies. And last but not the least, our participants, also my own mother, who passed away due to the Alzheimer's, and she was the source of my inspiration for this study. And also thanks to all my team members and volunteers who make this possible. I would be happy to receive your question if you have any. You can find my email by just Googling my name and Manitoba. Thank you.
Great, Professor. Outstanding presentation. Up next, we proudly present to you viewers an innovative line of research. It is the electrovestibulography, or EVESG, as designed by yet another bioengineering professor at the University of Manitoba. Its applications range from diagnosing neurological disorders to helping measure how effective a certain therapy would be in treating them through the use of wavelet decomposition. Give it up for Professor Brian Lith.
Good afternoon, all. I'm hoping you can hear and see me. What I'm going to do is talk to you about how we might quantitatively measure depression. And the technique I'm going to use is a thing called electrovestibulography. Now, electrovestibulography is a measure of sensory performance, and in particular, the sensory performance of the vestibular system. So the first question one should ask is, why should the balance system be different in depression? Well, this is a slide I like to show, which shows that fish